Proteomics analysis. Proteomics analysis refers to the systematic identification and quantification of the complete complement of proteins of a biological system at a specific point in time. Proteomics research permits the discovery of new protein markers for diagnostic purpose and the study of novel molecular targets for drug discovery. The principle is that functional groups of regions within the protein absorb light in the ultraviolet or visible range of the electromagnetic spectrum to 200 to 800 nanometers. Mass spectrometry has been widely used to analyze biological samples and evolved in indispensable tool for proteomics research. The types of protein test. Following the test carried to find the presence of proteins in a given sample. Burette test, xanthophoretic test, Marian's test, inheritance test. The presence of protein tested by the Pyrrhus test for proteins. The burette range made of sodium hydroxide and copper sulfate helps in determining the presence of protein in sample. This test and color test used to detect the presence of proteins in a solution. Only proteins having alpha amino acids give a positive inheritance test and a bluish purple color complex is formed. The secondary structure prediction. Secondary structure prediction is a set of techniques in bioinformatics that aims to predict the secondary structure of proteins and nucleic acid sequence based only on knowledge of the primary structure. Protein secondary structures predictions aims to construct a function that can map the amino acid sequence into secondary structures so that the protein secondary structures can be obtained according to the amino acid sequence. The method of secondary structure prediction. Statistical method, nearest neighbors method, neural network method, support vector machine method, and HMM method. Homology modeling. Homology modeling is one of the computational structures prediction methods that are used to determine protein 3D structure from its amino acid sequence. Homology modeling, also known as comparative modeling of protein. Homology modeling, also known as comparative modeling of protein, refers to constructing an atomic resolution model of the target protein from its amino acid sequence and the experimental three-dimensional structure of related homologous protein. Development of homology model is a multiple steps process and that can summarize in following way: identification of template, single or multiple sequence of alignment, model building for the target based on the 3D structure of template, and model refinement, analysis of alignment, gap deletions, and etc. Application of homology modeling. Because it's difficult and time consuming to obtain experimental structures from methods such as X-ray, crystallography, and protein NMR for every protein of interest. The homology modeling can provide useful structural method for generating hypotheses about a protein's functions and directing further experimental work. Thank you.